my name is Evie Salchuk, and I have been cultivating algae for the past 3 years in order to produce an economically feasible biodiesel. My first year, I tested algae under the nutrients phosphorus, nitrogen, and potassium, and I learned when grown under phosphorus, it grows better. In the second year, I tested algae under hydrostatic conditions versus ambient conditions, and learned that when grown under hydrostatic conditions, it absorbs more CO2 and grows to a greater quantity. Now this year, I didn't want to just focus on the cultivating stage, but instead the cultivating, harvesting, and extracting phase. Because right now, algae oil is not a contender in the biofuel race, because it costs too much to produce and it's not 100% green. So I wanted to focus on all three processes. When I first got my algae sample from Wards, it is star jar algae. I put it into my incubators. After growing in the incubators for 4 weeks, I transferred the algae into my vessels. Now these vessels, I made them out of polyvinyl chloride. I drew the drawings on AutoCAD and had them sent out to be machined, and when I got the parts back, I put it together. Right here is my CV chamber, controlled variable chamber, which is just a retrofitted Ward case. As you can see, it's a closed loop system. The CO2 gets pumped through the fish pumps into the vessel through the sparger, which is different from last year's before. As you can see from years before, it had large holes, so I had large bubbles of CO2. But I realized that the algae could not absorb the optimal amount. So instead of drilling all the way through, I poked holes with a pin, so there will be small bubbles of CO2, which will be absorbed more by the algae. When the CO2 bubbles up, it goes back into the CV chamber. Now, what I did was I had a red light spectrum photobioreactor versus a visible light spectrum photobioreactor. The red light spectrum photobioreactor and the visible light spectrum photobioreactor are the same, except that the red light spectrum photobioreactor has a prism, which I use to separate the light spectrums. And as you can see, only the red light went through the slot. And both the red light spectrum bioreactor and visible light spectrum photobioreactor have the same slot, so the same amount of light gets through. The reason I used red light is because it's of a higher electromagnetic frequency, 650 to 1,000 nanometers. Now, what happens is in algae cells, a chemical called ATP, adenosine triphosphate, allows for the conversion of CO2 and H2O into glucose, which is the fuel for algae cell, and through ATP. So what happens is when under red light, it speeds up this process, allowing more food to be created, so the algae grows to larger quantities. Now, when I transfer my algae solution into my vessels, first I allowed only distilled water to be my vessels, so it get acclimated. Then I pumped CO2 through to make sure my sparger works, and then I added the algae sample. When I added the algae sample, I took a hemocytometer test count, which I take a sample from my solution and put it on a Newborn grid underneath the microscope, so I can count the cells in a 1 millimeter by millimeter square. Then I took a CO2 airborne test. Each week I bring my CO2 baseline up to 650 ppms, so I can see how much was absorbed every week. And after 6 weeks in my vessels, as you can see, the red light spectrum photobioreactor started to grow more, and it also absorbed more CO2 each week by a greater amount, supporting my hypothesis that when in the red light spectrum photobioreactor, algae cultivates to a larger quantity and absorbs more CO2. Now, also while in my vessels, I inserted some iron through this porthole over here. And the reason for that is threefold. One, iron is a nutrient. Therefore, it increases the growth rate of algae. Two is that iron, when absorbed by algae, iron allows for the absorption of more CO2. And I learned that from Kenneth Cole, an Australian scientist who dumped iron mesh into the Pacific Ocean to create an algae bloom to absorb CO2 out of the environment. And after 6 weeks in my vessels, I no longer inserted any more CO2. And that was to lower the pH for the next step of flocculation and also to stress out the algae to produce a higher lipid count. So what I did was to flocculate my algae, I put some samples into test tubes. And the reason I used test tubes was because I wanted to narrow my focus so I can see what was happening. And what I saw was there's hydrogen coming out of my algae solution. And that's because of the flocculation process. The iron I added in during my cultivating stage comes into a second part during the harvesting stage. What happens is the Fe2+ ionically bonds with the oxygen in H2O, forming Fe2O3, iron oxide, which acts as a polymer, and through Coulombic interactions, van der Waals, dipole-dipole, and hydrogen bonding, it clings onto the algae cell wall, forming matrices that then create the algae to be denser and sink to the bottom. I flocculated out my whole vessel, and what I had left was an algae slurry. And as you can see from these two pictures, this is the algae when I first put in the test tubes, and 4 hours later, it was flocculated out. What I would do is the solution on top, I dewatered it using a pipette, and then recycled it and put it back into my incubators. It's a good way because I'm not using any chemicals that are harmful, so I don't have to post or pre-treat it, so it's easily recycled. And that's one good reason of my harvesting process. So after flocculating out my algae solution, I had an algae slurry. I ran it over an Amindium magnet, which is 7 times stronger than a normal magnet, to remove any excess iron mesh. Now with my clean algae slurry, I put it into my ultrasonic vacuum. No one has ever created an ultrasonic vacuum, and I wanted a way to remove the lipids, which are the oils to create a biodiesel, from my algae cell without the use of hexane, which is an expensive chemical currently at $58 a gallon, which is way too outrageous. So I came up with this idea because I knew ultrasonic machines are used in water treatment plants to kill bacteria by shredding it to pieces, and I wanted no outside constraints on my algae cell wall, so I used a vacuum. When I put my clean algae slurry into my ultrasonic vacuum, first I drew the vacuum to 100 millimeters of mercury, and that was to stress out the algae to produce even a higher lipid concentrate. Then I drew it to 600 millimeters mercury and turned on my ultrasonic machine to 1 kilohertz. As you can see, my ultrasonic probe is triangular while my beaker is circular. And that's because I want the sound waves to bounce off at all different directions, hitting the algae cell at different angles. Now, when in my ultrasonic vacuum, it creates areas of high and low pressure. During low pressure, cavitations are formed, which are bubbles that look like red blood cells. During the areas of high pressure, these cavitations implode, causing a jet stream that shreds through my algae cell wall, releasing the lipids that then float to the surface. After 10 days in my ultrasonic vacuum, you could see the lipid content on the top, therefore supporting that through my ultrasonic vacuum, you can remove the lipids without the use of hexane from an algae cell. So when you use all 3 of my processes, you can significantly reduce the cost of algae oil, therefore making it a feasible biodiesel.